Much. Thank you to Marco and Stefano for inviting me to this special session. And I'm going to talk about some techniques we've developed to work with large amounts of epigenomic data. So as a brief introduction, I'm sure many people here are familiar with the ENCODE project, but for those who aren't, the ENCODE project is something the NIH started about a little more than 10 years ago to try to figure out what sort of gene regulatory behavior was occurring in the genome-wide, using every assay that they could throw at it. So there have been dozens and dozens of papers that came out of ENCODE now, and I'll talk a lot about some of the data that was produced during the second phase of the ENCODE project. And a lot of this data falls into the general category of functional genomics. Right, so here you can see a chromosome, and you can see zooming through several different layers of magnification, from the whole chromosome in metaphase down to chromatin wrapped around in little beads on a string, so wrapped around nucleosomes that are then made up of nucleosomes are made up of histone proteins, which can have various modifications. You can zoom in further and see the actual DNA bases. And this structure is actually meaningful for both determining and also as evidence of what sort of gene regulatory behavior is occurring. And we now have wonderful assays that will allow us to measure a large number of these things. So for example, we have techniques like DNase and ATAC-seq that will tell us where regions of open chromatin are, where can the transcription factor machinery get access to DNA and transcribe a gene. We have techniques like ChIP-seq, which can tell you which transcription factors occur at a particular location, and also which histone modifications occur. So all these histones that the nucleosomes are made up of, they have big floppy tails, and there are post-translational modifications that can occur there, things like lysine monomethylation. We all use this very terse shorthand H3K4ME1 to refer to something like that. And so in the end, ENCODE produced some RNA-seq data as well, and we could get a great map in a lot of different cell types, a lot of different human cell types and tissues of where all the genes are and which genes are expressed. But also we have map of where cis-regulatory elements are, things like promoters, and long-range regulatory elements like enhancers. So there was a lot of different data produced from a lot of different groups. So you can represent it in this sort of hinge format where the y-axis is the number of different cell types, the x-axis is the number of different assay types, things like ChIP-seq, DNA-seq, and so on. And for ChIP-seq specifically, there are now in the ENCODE database 710 different individual targets. Some of them are slightly redundant with each other, so there aren't really that many individual transcription factors we have, but still, it's a lot of different data. In ENCODE, as of a few weeks ago, there were more than 10,824 experiments deposited, and the question when you're faced with this much data, all of it is genome-wide, dense, high-resolution data, now what do you do with it? So I was part of the analysis group for ENCODE, and we developed a technique called semi-automated genome annotation. And semi-automated genome annotation, we take the sort of signal data, and I'm going to describe exactly what that means in a second, that you get from these sorts of data sets, take multiple tracks of signal data and discover patterns in them that we can then use to annotate the genome, so turning very complex, multivariate signal data into simple annotations that a human can understand, visualize it as well. So this is also a simplification process to make it easier for a biologist or other analyst to understand what's going on, and also to interpret other data sources we have. So if we have information from, say, a GWAS or some other genetic information in a non-coding region, and we want to figure out why it is affecting the genome, what is the functional effect, you can use something like the techniques we have developed. And many other techniques are actually built on the annotations we produce, which which is another story, I suppose. So let me talk a little bit about semi-automated genome annotation. We do it specifically through a genomic segmentation task. And what we do is we take a number of data sets, where here I'm showing 3, and on the x-axis is position in the genome, and the y-axis is the signal. The signal for a, experiment on a population of cells is a function of the number of cells that show some property at that location. Okay, so if you did 10 million cells and some of them have open chromatin at some particular location, and some positions, some of them have more, you'll get a little bigger peak than over at the place where you have some less. So that's how you get this signal, and that's what it represents quantitatively. So in the genomic segmentation task, we partition the genome into a number of non-overlapping segments, and then we give each segment a label from a finite set, and we change the locations of the segments and change the identities of the labels to maximize the similarities between segments with the same label. So here you can see, for example, a 0 label with a low-high-low pattern, a 1 label with a high-low-high pattern, a 2 label with a low-high-high pattern, and so on. All right, so really, in essence, it's a simultaneous segmentation and clustering technique. You don't do them separately, you do it at the same time to kind of maximize the likelihood of the model we have, which I won't go into in depth, but is described in the paper. So all of this is implemented in our software, which is called Segway, because it's a way to segment the genome, and we have continued development of Segway, and it is all available for free for you to use, and I'll talk a little more at the end about that. So I just wanted to show you a couple of examples of how we've used Segway. So here's one example where we took 49 different data sets from ENCODE. They're all from the same cell type, K562, which is a chronic myeloid leukemia cell type. And then we told Segway that we wanted it to find 25 labels, 25 different patterns or states within this data. All right, so this question here of how many labels do you pick, it's really a trade-off between how many different kinds of behaviors you want to see and how easy it is for a human to understand. So you could pick a number like 200 and there would be enough statistical support for it, and then it would be utterly useless because no one would be able to remember the difference between state 170 and state 36. So in the end, after doing that, after running the training mode of Segway, we get a set of emission parameters. All right, so this matrix shows all of the emission parameters. It's a matrix of 25 patterns along the x-axis times 49 different data sets. All right, and everything is row normalized. So any row where you have the reddest value, that indicates the highest mean that has been learned. So we learned 49 times 25 different Gaussians, and you can see all the means there. Those are the colors. We also figured out a way to represent variance as well with these tiny little black bars in the middle. So already, so after doing this, we've hierarchically clustered the parameters just to make visualization easier, and you can already see, I think, kind of 3 major groups at the top. You have this group over at the left, where there's a lot of blue, and then there's a group in the right where there's a fair amount of red in the top block and some going down, and then kind of a group in the middle. And when I described this earlier as semi-automated annotation, this is everything up here has been automatic, but this is the semi-process where someone with a large amount of domain knowledge, in this case it was me, went back and took these numbers 0 through 24 and replaced them with mnemonic hypotheses that better describe what these things actually do. So we have these little abbreviations followed by numbers, and you can already see here that 25 is maybe too many patterns because we have some very similar things that we distinguish by numbers. So these sorts of things that we will find are things like transcription start sites, a little bit further into the gene, which I'll call gene start, gene middle, 3' ends of genes, distal enhancers, distal CTCF or insulator regions, polycomb repressed regions, and then these so-called dead regions. Okay, so the dead regions, indicate one of the things you can get out of using an unsupervised method here. No one really, and looking at all of these different data sets simultaneously as well, no one really expected this before we did this, but there are a lot of regions that in some cell type, every chromatin assay comes up with very low results, right? So, and it's not a sequencing artifact, because where these positions are changed from cell type to cell type, and only by looking at all these together and letting the system do unsupervised analysis, people realize there's actually, this is actually an interesting and reproducible phenomenon. We call them dead regions. Unfortunately, within the ENCODE consortium, dead was rather unpopular. So if you look for this in the published paper, you will find it as quiescent. So just so you know. So using a little bit of a more recent segmentation model, I can show you how this works. After you do that training, then you can go back to the genome and you can give a label to every position in the genome. So this sort of gestalt of which pattern does every region map best. So here you can see what looks like a bidirectional promoter. Okay, you can see the gene track in the middle with two genes that are diverging. And then below, you can see a number of the individual tracks that were used in the segue analysis, two big histone marker-related signals on either side, and kind of a signal of DNA accessibility in the middle, right? And at the top, you can see the segue annotation, and segue has called that part in the middle regulatory / open chromatin, and the part immediately beside it is transcription start site, and then promoter flanking, and so on. And there are a lot of technical tricks that we used in order to get segue to run at 1 base pair resolution and use the full resolution of this data, which is the only reason you can actually distinguish between these two things. If you downsample it and use much bigger regions, a lot of this stuff becomes technically a lot easier, but you can miss some of these fine details. If we zoom out a bit and look at another 20 kb region of the genome, and I'm going to change kind of the visualization here so each possible pattern has a row of its own, and when it gets thick, that's where you actually find that. And we can find, these TSS gene start, gene end patterns occur all throughout the genome. All right, so this is something, in a totally unsupervised fashion, this essentially rediscovered genes, and we didn't use anything that people traditionally use to find genes, so there was no comparative data, there was no RNA data, there wasn't actually even any sequence data whatsoever. There's only this signal, this numerical transformation of this ChIP-seq data, and we were able to find this. And you can find the same thing in bigger regions as well. So then we started, this worked all throughout the genome, and in aggregate, it looked really good, but it's always really important as a bioinformatician to always look at a lot of individual examples, because just looking at the average can really mislead you sometimes. So we started doing that, and then we noticed this region where we have all these genes, right, and segue calls like one gene here, and then all of the other genes, it keeps calling these dead segments, these dead labels, these and other things, no GS segments. And the reason, of course, is that those genes were not turned on in this cell type. So it's more than just finding the genes, it's genes that are actually active in this cell type, amongst other things. So we decided to see whether we could use the predictions of segue to find those TSS-associated sequences that are sufficient in and of themselves to drive transcription in this cell type. All right, so we hired a firm, Switchgear Genomics, to do our experiments for us. Like some people do their bioinformatics in the cloud. We do our wet lab biology in the cloud, and we just send off spreadsheets to them, and they send us back the results. And basically, we found that every single one of the predictions that we made as a positive prediction, that sequence was sufficient to drive transcription. And the negative predictions, a majority of them showed no expression. There's less expression overall. And it must be that in these cases, they're all TSS-associated sequences. They do not repress in this cell type. Something else around them is actually causing repression. So our collaborators also did some amount of testing of predictions we made for promoters and transgenic Medaka fish. So all of this stuff I've shown you is from acute myeloid leukemia cells, and they tested in transgenic Medaka fish in Heidelberg. And what you can actually see here these glowing green cells. Medaka have nucleated red blood cells. So we actually have the GFP reporter in those blood cells, and you can actually see these glowing red blood cells moving through the Medaka fish. And we also did some tests of our enhancer predictions and compared against other groups within ENCODE. So a couple of other groups doing discriminative analysis compared to our unsupervised graphical model approach, right? And then naive approach based on accumulation of transcription factor binding sites, and then the so-called biologist approach, where a trained principal investigator of several decades of experience kind of eyeballed the UCSC genome browser and said that's an enhancer. And not surprisingly, our method and the computational methods did much better. Well, it didn't surprise me. I think it surprised the biologist. And I'm running a little short on time, but you can see this in our NAR paper where we show how you compare segue annotations against things in the GWAS catalog, and you can find that those phenotypic changes in the GWAS catalog have a big excess over random mutations you might find somewhere like 1,000 genomes that may or may not be phenotype associated. This big excess associated with the segue enhancer pattern, the segue enhancer flanking pattern. And the third biggest category is this REPR2, second repression. So I think these may represent essentially loss and gain of function of gene regulatory activity, but people need to look into that a bit further. So just in case you want to use this yourself, segue.hoffmanlab.org, you can download the software and run it. And I've had a professional programmer working on it for a while, so it's a lot easier to install than when I was a postdoc. There's also all the annotations that we've produced that you can download and not run the software, just download it, use your favorite tool, or you can click where I have circled in red, and then you'll load the segue tracks into the UCSC genome browser. They're built into Ensemble as part of the Ensemble regulatory build, and we continue working on more of those things. So just to sum up, segue, which is kind of semi-automated genome annotation, is a technique that you can use to find patterns in genomic signal and use those to annotate the genome in a human understandable way, visualize complex multivariate data in a simple way, and interpret things such as genetic variation or cancer somatic mutations. And I hope you'll check out the software. So I'd like to thank everyone in my lab, a lot of people who have helped during this work, and finally I'd like to thank all of you for your kind attention. Thank you very much.